Good day, students. This is Julian Reses Leroy from the Department of Plant. But before we proceed to the questions, here are the guidelines. This is your first question. Which of the following statement defines crop improvement or plant breeding? A. It is a science of improving the genetic makeup of a plant population for the benefit of mankind. B. It is an application of genetic analysis to develop varieties better suited for target environments and human needs. C. It is a science, art, and business of improving plant for human benefits. And D. All of the above. Your timer starts now. Time is up. Okay. The answer is letter D, all of the above. All the statements defines plant breeding or crop improvements. Plant breeding is a science which applies genetic analysis to improve the genetic makeup of the crop or plant population to become suitable for different target environments and to cater human needs. It is an art since it involves creative imagination to express the technical proficiency and conceptualize ideas. Plant breeding also is a multi-billion industry in both local and international market. Thus, plant breeding is also a business. This is your second question. What term defines as the hypothetical optimum partitioning of dry matter according to the purpose of which the cultivar will be used? A. Yield potential. B. Plant idiotype. C. Biological yield. D. Economic yield. Your timer starts now. Time's up. The correct answer is plant idiotype or idiotype. So how about potential yield? What is potential yield? This is the inherent optimum capacity of a given crop to perform under a given environment. Whereas biological yield, this is the total dry matter per plant or per unit area. While economic yield is the total weight per unit area of a specified plant product which has economic value. So again, the answer is plant idiotype. Your third question is, which of the following is not a breeding goal in crop improvement? A. Pest and disease tolerance and resistance. B. Abiotic stress tolerance. C. Improve crop nutritional and industrial quality. D. Increase yield production. E. Improve plant traits suitable for mechanized agriculture. And F. None of the above. Timer starts now. Okay, time is up. The correct answer is letter F. None of the above. All of these statements are possible breeding goals in crop improvement. For example, plant breeders aims to produce pest and disease tolerant and resistant crop. When you say plant tolerant crop, this is the ability of the plant to reduce the effect of infection on its fitness regardless of the level of pathogen multiplication. Whereas plant resistance means that the plant able to reduce the growth or presence of the pathogen. Another breeding goal is the development of tolerant crop tolerant to different environmental conditions such as high or low temperature, high or low light, high or no water, or low water, or when you say flooding or drought, and then high or low CO2 level and other environmental factors that can affect plants. Also, plant breeders wants to cater food security and nutritional deficiency through developing high-yielding crops which are rich in vitamins and other nutrients. For industrial and mechanized production, plant breeder also develops crops suitable for these types of production. So again, the answer is none of the above. This is your fourth question. Which of the following is the correct sequence of the basic steps in crop improvement? Here are the basic steps of crop improvement in no particular order. First, you have collection of variable accession, evaluation of and selection of possible parents, hybridization, evaluation and selection of superior recombinants. Last, you have testing, release, and commercialization of new cultivars. And now, here are your options. For letter A, it starts with collection of variable accession, followed by evaluation and selection of possible parents, followed by hybridization, followed by evaluation and selection of superior combinants, and last but not the least, testing, release, and commercialization of new cultivars. Option B, you have evaluation. It starts with evaluation and selection of possible parents, followed by collection of variable accession, followed by evaluation and selection of superior recombinants, followed by hybridization, and last but not the least, testing, release, and commercialization of new cultivars. For option C, it starts with evaluation and selection of superior combinants, followed by collection of variable accession, followed by hybridization, followed by testing, release, and commercialization of new cultivars, and last but not the least, evaluation and selection of superior recombinants. For the last option, option D, it starts with testing, release, and commercialization of new cultivars, evaluation and selection of superior recombinants, hybridization, next, evaluation and selection of possible parents, last but not the least, collection and of variable accession. Your timer starts now. Time is up. The correct answer is letter A. It starts with collection of variable accession. So first you have to create variability, and then followed by evaluation of possible parents. So out from that variability, you have to select possible parents for your next step, hybridization. So these possible parents must have the traits that you want to develop. And then you hybridize in the third step. After that, after hybridization, you will be able to produce progenies. So in the fourth step, you have to evaluate and select superior recombinants or your progenies. After selecting the superior recombinants and progenies, these superior recombinants and progenies are tested or sent to testing centers, sent to different field trials. After several testing or after several field trials, this will be released. After release, it can be commercialized and serve as your new cultivar. Okay, so that is the correct sequence of the basic steps of crop improvement. Again, collection of variable accession first, second, evaluation and selection of possible parents to be used in your third step, which is hybridization. And then after producing progenies, you have to evaluate and select superior recombinants or progenies. After that, you have to test, release, and commercialization of new cultivars. This is your fifth question. Which of the following is not considered as domesticated plants? A. Commercial cultivars. B. Breeding materials. C. Landrace. D. Plant introduction. And E. Wild plants. Your timer starts now. Time's up. Okay. Before we proceed to the answer, let us first define what is domesticated plants. Based on the definition, domesticated plants are those plants that have been subjected to some form of human selection, or these plants are selected by humans because of some important purposes, which humans think that they are important to them. For example, if they are source of food, source of fiber, source of medicine, and other sort of importance to humans. So, what are these plants? These are plants cultivated by humans for food consumption, for aesthetic purposes. So, when you say aesthetic purposes, the plants grown by your titas, plantitos and plantitas, those are domesticated plants. Now, you have idea now which of the option is not considered as domesticated plants. Okay. So the correct answer is wild plants. Even the wild plants or the weedy relative of our domesticated plants are important in the breeding program because they become source or donor of important disease and insect resistant genes or genes for adaptation, but yet they are still wild. They are not yet domesticated. Okay. So how about commercial cultivars? So what are commercial cultivars? So commercial cultivars, these are cultivars that are product of formal breeding program. So there are two forms of commercial cultivars. You have the current cultivars and the obsolete cultivars. So for the current cultivars, these are the cultivars that are still produced commercially or still available in the market. Whereas the retired or obsolete cultivars, these are previously cultivars and they are taken out from the commercial production because of susceptible to disease or they become low yielding because there are more varieties released that are high yielding compared to that obsolete cultivars. Okay. Next, breeding materials. So what are these? These are intermediate breeding products are usually genetically narrow based because they originate from a small number of genotypes or population. So this also includes ongoing or more established breeding programs, maintains variability from previous projects. Okay. Next, landrace. What are landrace? So landrace cultivar, these are farmer developed or maintained cultivars. So the farmers develop this or maintain. So this includes your heirloom cultivars. They are developed over very long period of time and have co-adapted gene complex. Okay. So these cultivars are passed from one generation to the other generation. So that is a landrace. Okay. Next, plant introduction. These are new plant materials may be evaluated and adapted to a new production region as new cultivar or used as parents for crossing in breeding projects. For example, we import some plant cultivars from Indonesia and we introduce that here in the Philippines. So that plant that is called plant introduction. Before that is not available in the Philippines and now since you import it, now it becomes available to the Philippines. Okay, so that is called plant introduction. Okay, let's proceed to the next question. This is your sixth question. Which of the following event is not a result of genetic vulnerability? A. 1845's late blight disease of potato in Ireland. B. 1970's southern leaf blight disease of corn in the United States of America. C. 1980's hybrid maize failure in Zimbabwe. And D. None of the above. Your timer starts now. Time's up. Before we proceed to the correct answer, let us first define genetic vulnerability. It is a term used to indicate the genetic homogeneity and uniformity of a group of plants that predisposes it to susceptibility to a pest, pathogen, or environmental hazard of large-scale proportion. So meaning a population having uniform genetic background, and that genetic uniformity in the genetic background makes them vulnerable to some pest, pathogen, and environmental hazard. For example, if a plant coming from that population becomes susceptible to a certain disease, automatically the rest of the population will be susceptible too because they have the same genetic background. And if that disease is lethal or very virulent to to that genotype, and there is a chance that that specific genotype will be wiped out. In worst case scenario, if that is a species, there is a chance that that certain species will be wiped out. Okay. So now let's go back to the correct answer. And the correct answer is letter D, none of the above, because all of these events are results of genetic vulnerability. For example, the 1845's late blight disease of potato in Ireland is a result of monoculture of narrow genetic base potato in Ireland, and it became susceptible to the late blight disease in 1945. So by the way, Ireland, their staple food there is potato. So they planted lots of potato, monoculture of potato. Okay. And this certain genotype they use is having a narrow genetic base. So there is no variability on that sweet or on that potato they planted in their field. And here comes the late blight disease in 1845. And that certain genotype is very susceptible to that late blight disease. And this caused famine in Ireland because all of their potato fields became wiped out because of that blight disease. Okay. Next, you have here 1970 southern leaf blight disease of corn in the United States of America. This genetic vulnerability in corn was attributed to the genetic uniformity due to the widespread of the T cytoplasm in hybrid corn seed production. Unfortunately, this hybrid corn with T cytoplasm background is susceptible to the new strain of H. maydis surfaced around 1970, which caused devastating consequences due to the large acreage of the crop. Okay. And the last but not the least is the 1980s hybrid maize failure in Zimbabwe. So the genetic vulnerability of these in Zimbabwe is due to the narrow genetic base of hybrid maize as compared to the traditional cultivar in Zimbabwe in response to the drought in the mid 1980s. Okay, so all of these events are results of genetic vulnerability. This is your seventh question. Which of the following step is not included in the procedure of collecting plant genetic resources? A. Logistic and technical planning. B. Collecting plant sample or planting material. C. Gathering passport information. D. Documentation. E. None of the above. Your timer starts now. Time is up. But before we proceed to the correct answer, plant genetic resources is the first step in your breeding program. You have to collect different genotypes of your crop of interest so that you have variability for your breeding program or breeding plan. Okay, so now let us know the correct answer. And the correct answer is letter E, none of the above, because all of these steps here are included in collecting plant genetic resources. For example, logistic and technical planning includes formulation of team members or personnel. So who are the members of the team or collecting team? So the members of the collecting team must be a plant expert. So if you are collecting agronomic crops, you must have agronomists. If you are collecting horticultural crops, you must include horticulturists in your group or in your team. And most of all, you must have a plant breeder because the plant breeder will be the one using those genetic resources. And also, you must have a soils expert because the soil expert will assess the area where your plant genetic resources came from. Also, pest and disease expert. Not only them, but also extension specialist. So, for example, the plant and disease expert, they will assess in their the area where the plant grows or where you collected your plant genetic resources, they will assess if there is a presence of disease or a pathogen or pest there present in their nature. Okay. And then also extension specialists because they will be the one coordinating with the locals where you collect your plant genetic resources. And again, logistic involves coordination of local indigenous people folks, accommodation, transportation, and other legal matters of the trip. Okay. Next, collecting plant sample involves collecting viable plant propagule to be planted in the collection nursery. C. Gathering passport information. So passport information includes the collection information, initial plant description, other significant information about the collected plant. And last but not the least, documentation. This includes photographs of standing crops on the field of origin, signing of agreements, and other important events during the trip. Okay. So again, the correct answer is letter E, none of the above. Question number 8. Blank is a breeding concept that facilitates the transfer of several resistant genes simultaneously into one genotype. A. Multiline breeding. B. Composite breeding. C. Gene pyramiding. D. Synthetic breeding. Timer starts now. Time is up. And the correct answer is letter C, gene pyramiding. Okay. Gene pyramiding is a breeding concept or scheme that facilitates transfer of several resistant genes coming from different parents simultaneously into one genotype. So it involves several parents having these resistant genes. Then you will do several crossing procedures to be able to transfer the genes into one genotype. Okay. On the other hand, multi-line and composite breeding involves seed mixture. Multi-line use several cultivars or lines developed from different block crosses or back crosses procedure such that each component or each cultivar in the mixture constitute at least 5% of the whole mixture. Each pure lines are phenotypically uniform in terms of agromorphological traits. Whereas composite breeding, as I've said earlier, it's also involved mixture of different inbreds, hybrid lines, population, which are having less similar genotypes. So they have different genetic background, but they are similar in terms of growth periods, degrees of resistance either to lodging or pests and diseases, and this mixture is released as a variety. On the other hand, synthetic breeding involves several generations of cross-pollination among a number of selected inbred lines or parental cultivars having promising genetic background, and then the resulting population will be released as an open pollinated variety or OPV. Okay. So again, the correct answer is letter C, gene pyramiding. Question number 9. Which type of cultivar is produced from crossing selected inbred lines coming from different heterotic groups? A. Pure line. B. Open pollinated variety. C. Hybrid cultivar. D. Clonal cultivar. E. Apomictic cultivar. Your timer starts now. Time is up. And the correct answer is hybrid cultivar. So hybrid cultivar is produced from crossing selected inbred lines or pure lines coming from different heterotic groups. Okay. So again, the answer is hybrid cultivar. So the remaining option, we will define the remaining option. So first, you have the pure line or inbred line. This is produced from successive selfing of naturally or cross-pollinated crops until it reaches a certain level of genetic homogeneity or homozygosity. And OPV or open pollinated variety is produced from naturally crossing pollinated crops, which is genetically heterogeneous and heterozygous in nature. So we have mentioned that OPV or open pollinated variety usually develop from the synthetic breeding scheme. Okay. And letter D, you have the clonal cultivar is produced using vegetative propagation of previously improved or developed crops. Thus, the clonal cultivars and the parental materials are genetically identical. So clonal cultivars, these are vegetatively propagated from from previously developed crops. So for example, one basic example of this one is your cassava and sweet potato. So cassava, you develop a hybrid cassava, and then in the next generation, you will produce or you will release it as a clonal cultivar since you only need the stem of the cassava to release it as a variety. Okay, the stem of the developed cassava variety. Okay. And last but not the least, the apomictic cultivar. So apomictic cultivar is utilize the phenomenon called apomixis or seed production without fertilization. Thus, apomictic cultivar and the parental lines are genetically identical. So same with your clonal cultivar. In this case, in clonal cultivars, all plant parts except the seeds that you will release as variety. But here in apomictic cultivar, it is the seed, but this seed is produced from the phenomenon called apomixis. So seed production without fertilization. So basically, that seed, the genetic makeup of that seed is equivalent to the parental material. So they are genetically identical. In this case, seed lang siya, liso siya. Okay. So again, the answer for this question number 9 is hybrid cultivar. Question number 10. Which of the following breeding method aims to improve the population by increasing the gene frequency of the desired gene in the base population of a naturally self-pollinated crops? A. Mass selection. B. Pure line selection. C. Pedigree selection. And D. Bulk selection. Your timer starts now. Timer's up, and the correct answer is A. Mass selection. So here, you have here a schematic diagram of mass selection. So you have here, in the first year, you have the source population. It consists 500 to 100 desirable plants. And then in the second year, out from the source population, you only collect or select 200 plants and then plant it in heads in row. And then you have to rogue the off types or remove the weeds and other not included plants. Then after that, in the year 3, you have to bulk harvest the selected plants. Okay, so that is mass selection. Okay, and here is a pure line selection. So pure line selection involves a repeated cycle of selfing following the initial selection of mixture of homozygous lines. This was developed by a Danish botanist in 1980, 1930, named Johansen. Okay, so here in the first year, you have 1,000 plants. Okay, and then in the second year, out of that 1,000 plant, you only select 200 plants and plant it in plant progeny rows. Then next, in the third and fifth year, in every year, you have to select only 25 to 50 plants, and that selected plants is planted in rows. And again, in the sixth year, you have to select only 15 plants to be included in the preliminary yield trials. And then in the seventh or 10th year, you have your advanced yield trials. So out from the 15, you only select 10 for the advanced yield trial. And then last but not the least, you have to select 2 or 1 as your release variety for this pure line selection. Next will be the pedigree selection. So the pedigree selection is a widely used method of breeding self-pollinated crops where hybridization is used to generate variability. This is applicable to breeding species which allows individual plants to be observed and described and harvested separately. So here you have your hybridization part in the first year, and then you have to plant your F1 hybrids, that is 50 to 100 F1 hybrids. And then in the third year, you have to plant the progeny of your F1 hybrids, that is F2. And then you have to select only, so in the F2, you have 2,000 to 5,000 F2s. You have to plant that in the field. Then after that, you have to advance only 200 of that F2s. So meaning advance, you have to select only 200 and plant it in the following year. And then you do selection. So you have to select plant in space rows here. So you have first selection. Your first selection will be during the third year up to, you have to do selection up to the 8th year, and also in the 8th or 9th to 11th year. So you have to do selection here in the here process. Then last but not the least, after the 11th year, you have to produce your cultivar, a pure line cultivar or an inbred line. Okay. Next, Last but not the least, you have your bulk selection. So bulk selection is a breeding strategy which takes advantage of the effect of the natural selection in the earlier generation by delaying the stringent artificial selection until the later generation. So here, this is a schematic diagram or procedure for your bulk selection. So you have here in the first year, you have your generation of variability, so your hybrid process. Then you create you produce your F1 and then plant the F1 in the following year, in the third year, in the second year rather. And then advance your F1 in the third year, so you have your F2 here, and then until you reach F6. So in the sixth year, there you have your plants. So you have many plants because you don't select. From F1 to F6, you don't select. So you plant all those seeds, bulk, and plant the seeds. Then in the seventh year, you start your selection. In the later years, you start your selection. And then after 11th year, you produce one cultivar, one or two cultivar to be released. Okay, so that is for your bulk selection. Question number 11. Which of the following breeding strategy best suited to improve an established cultivar which is later found to be deficient in one or two specific traits? A. Single citizen. B. Backcross breeding. C. Recurrent breeding. And D. Mutation breeding. Your timer starts now. Time is up. And the correct answer is letter B, backcross breeding. So backcross breeding enables breeder to transfer a desired trait from one variety, or that is called the donor parent, into a variety with a favored genetic background, that is the recurrent parent. On the other hand, seeding single seed descent is a modified pedigree method by which single seed of each line produced is advanced to the next generation before the individual plant selection and evaluation begins. Whereas recurrent selection in the option letter C is a cyclical and systematic breeding techniques in which desirable individuals are selected in the population and mated from one to form new population. This procedure is repeated until the desired genetic gain is reached. Last but not the least, mutation breeding is a process of exposing seed or any planting materials to chemical or radiation or other mutagenic agent in order to generate mutants with desirable traits to be bred with another cultivar. Question number 12. Which of the following recurrent selection procedure does not use tester? A. Phenotypic recurrent selection. B. Recurrent selection for general combining ability. C. Recurrent selection for combining or specific combining ability. And D. Reciprocal recurrent selection. Your timer starts now. Okay, time is up. But before we proceed to the correct answer, let us first define tester. So tester is a plant with known performance which is used to measure the performance of a certain crop or an evaluated crop. Okay. Now let's proceed to the correct answer. And the correct answer is letter A, phenotypic recurrent selection. Okay. So phenotypic recurrent selection is a type of recurrent selection in which the phenotype of the individual evaluated crop serve as the basis for selection. By the way, before we proceed to the other option, I have here a simple schematic diagram of recurrent selection procedure. So in here, this is a cyclic procedure. So in year 1, You have here a starting material. For example, you have here 20 elite lines, and then that elite lines will be crossed from each other. So that elite lines contains the gene of interest in your breeding program. And then you have to cross that to produce your progeny. And then in the year 2, your progenies from year 1 will be evaluated, and you have to select superior progenies. Some of these superior progenies can be advanced to your hybridization program. And then the rest of the superior progenies will be recombined or crossed again. And then the recombined progenies will now be selected again. The progenies of that recombination of your progenies will be selected, and that will serve as your elite breeding line or starter line for your second cycle of your recurrent selection. Okay, let's go back to other types of recurrent selection. Okay, so you have recurrent selection for general combining ability. This type of recurrent selection uses a wide genetic base tester such as your cultivar. And for the recurrent selection for specific combining ability, it also uses a tester with narrow genetic base such as your inbred lines. And last but not the least, your reciprocal tester, since it exploits both GCA and SCA, then both of the populations serve as the tester. So each individual in the two population were self and cross with the plants from the reciprocal female tester. So again, the correct answer is phenotypic recurrent selection. It does not use tester. Okay. Question number 13. Blank are landmarks on the chromosome that serve as reference point to the location of the gene of interest when a genetic map is constructed. A. Morphological marker. B. Genetic marker. C. Biological marker. And D. Molecular marker. Your timer starts now. Time is up. And the correct answer is B, genetic marker. Okay. So genetic marker, these are landmarks on the chromosome that serve as the reference point to the location of the gene of interest in the genetic map constructed or the constructed genetic map. Okay. How about morphological markers? Morphological markers are manifested with adult phenotype, which are the product of gene and environment interaction. So morphological markers, these are the traits that you can see or you can observe in an adult organism. And then for biological markers is a measurable indicator of some biological state or condition, which is often measured and evaluated using blood, urine, or soft tissue to examine normal biological processes, pathogenic processes, or pharmacologic response to therapeutic intervention. Whereas molecular markers in molecular biology or biotechnology, molecular marker is any molecule or chemical used to identify particular sequence of DNA in a pool of unknown DNA. So molecular markers are detected in submolecular levels and can be assayed before an adult stage of the crop. Okay, again, the correct answer is genetic marker. Question number 14. Which of the following is not used to detect molecular markers? A. Enzyme. B. Hybridizing fragment. C. Polymerase chain reaction procedure. And D. DNA sequence. Your timer starts now. Okay, time is up. And the correct answer is letter D, DNA sequence. Okay, DNA sequence is not used to detect molecular markers, but molecular marker is used to detect DNA sequence. Okay, for the other option, so in an enzyme-based marker assay, it uses enzyme to detect variation in the protein product translated from prospect DNA, not the DNA per se. Okay. Second, so in a DNA-based or hybridization-based marker, it visualizes DNA profile by using hybridizing fragment to label the probes. Okay. And last but not the least, the PCR-based marker includes amplification of a specific short segment of the DNA without using any cloning method. Okay, so A, B, C, the enzyme hybridizing fragment, PCR chain reaction, these are used to detect molecular markers. Okay, so again, the correct answer is letter D. Okay. Question number 15. Which of the following is not a PCR base marker? A. SSR, B. ISSR, C. RAPD, D. RFLP, E. DAF, F. SCAR, G. STS, H. AFLP, and I. SNP. Timer starts now. Okay, time is up. And the correct answer is letter D, RFLP. RFLP stands for Restriction Fragment Length Polymorphism. This is a hybridization-based marker which visualizes the DNA profile using labeled probes. So it doesn't use any PCR or polymerase chain reaction procedure. So it uses hybridization techniques. Okay. So how about the rest? What are the definition of those acronyms? Okay. So by the way, before we proceed to that, PCR or polymerase chain reaction is a technology for direct amplification of specific short segments of DNA without the use of cloning methods. So we have mentioned that earlier. Okay, and now let's proceed to the definition of those acronyms. So for SSR, that is stands for simple sequence repeats. These are repetitive DNA sequence in shorter motif usually found in the microsatellites of the chromosome. Okay. Then, so you have ISSR. This is enter-simple sequence repeats. These are also repetitive DNA sequence found in between microsatellites of the chromosome. And then for the RAPD, so that stands for random amplified polymorphic DNA, is a marker system which amplifies the total genomic DNA using a single short random primer. Okay. Then for the DAF, that stands for DNA amplification fingerprinting, similar to RAPD, but uses short primer. RAPD uses 10 BP or 10 base pair, whereas DAF or DNA amplification fingerprinting uses 7 to 8 base pair primer. Okay. Then SCAR, that is sequence characterized amplified region, sequence in the end of the RIPD fragment. Okay. Then the STS, that is sequence tag site, sequence at the end of the RFLP marker. And you have AFLP, amplified fragment length for polymorphism, basically basically RLFP visualized by selective PCR amplification of DNA restriction fragments. So this is the PCR-based component or equivalent of your RFLP. Okay. Then last but not the least, that is the single nucleotide polymorphism. This is a type of detection of the single base pair site in the genome that is different from one individual to another. Also uses PCR. So again, the following markers here or molecular markers here that does not use PCR procedure or polymerase chain procedure is the RFLP. Okay. Question number 16. Blank refers to the increase of overall productivity of a crop over the average performance of its parents or the better parent. A. Dominance theory. B. Heterosis or hybrid vigor. C. Overdominance theory. And D. Inbreeding depression. Timer starts now. Okay, time is up. And the correct answer is B. Heterosis or hybrid vigor. By the way, there are two types of heterosis or hybrid vigor. If the increase of the overall productivity of the crop is over the average of the better of performance of its parents, that is called mid-parent heterosis. But if the increase of overall productivity of the crop over the better parent, that is called better parent heterosis. Okay, so now let's define the other options. So dominance theory. So dominance theory, it assumes that the vigor in plant is governed by dominant alleles while the recessive alleles are being deleterious or neutral in effect. On the other hand, overdominance theory is a phenomenon of the heterozygotes being superior to the homozygotes. Last but not the least, inbreeding depression is the reduction of fitness as direct result of crossing closely related individuals. So again, the correct answer is letter B, heterosis or hybrid vigor. Question number 17. In intrapopulation improvement, what breeding method is represented by polycross mating system and ear-to-row selection? A. Full sieve family selection. B. Half sieve family selection. C. Self-family selection. And D. Individual plant selection. Timer starts now. Time is up. And the correct answer is letter B, half sieve family selection. By the way, class, polycross mating is used for cross-pollinated crops. And this is employed in the sweet potato breeding in Philippine root crops. So they have this polycross mating system. First, they select elite breeding lines or lines having their gene of interest. They plant it in the field in such a way that each individual plant has the opportunity to mate with the other member of the group planted in the polycross mating system. And then, after that, they allow cross-pollination or random mating. Then after that, if the plant bears fruit, they collect that fruit. And in the case of sweet potato, you can collect 2 to 3 or 4 seeds in per fruit. And then in that fruit, you are sure of the female parent because the female parent bears the fruit. So in each seed there, you only knew the female parent. The male parents you don't know, but you are sure that the male parent is coming from the other member of your plant in your polycross. Okay. So there, that seed is called half seed because you only know half of the parent, the female parent. In ear to row selection, this is employed in corn. So one ear, the seed of that one ear is planted in one row, or a composite of the seed in that ear is planted in one row. Also, one ear, in that one ear, there are several seeds there. You only know the female parent. But the male parent, you are not sure of which male parent fertilized that specific seed. So that is also a half seed family selection. Okay. How about full seed family selection? So full seed family selection is generated by biparental crosses using the parents from the base population. Okay. So when you say biparental crosses, you are sure of the parental or both parents of the cross. So you do artificial cross-pollination in this one. Okay. The next, for the self-family selection, this is generated by selfing the lines, evaluated the self-families in multi-environmental trials, and recombine the remnant seed of the selected self-families. So that is for self-family selection. And last, the individual plant selection, regardless of their uh, if you know the both parents or the single parents, if they are self parents, they are evaluated individually and they are selected individually based on their performance. That is for individual plant selection. Okay. Again, the answer for number 17 is letter B, Hapseed family selection. Question number 18. What Republic Act mandates the replacement of the Philippine Seed Board to National Seed Industry Council as the governing body responsible for the approval and registration of crop varieties in the Philippines? A. RA 7038, B. RA 7830, C. RA 7083, and D. RA 7308. Timer starts now. Time is up. And the correct answer is letter D, RA 7308. Question number 19. Which of the following is the first commercialized genetically modified crop? A. BT corn, B. Flavored savory tomato, C. Roundup Ready soybean, and D. Golden rice. Your timer starts now. Time is up. And the correct answer is letter B, flavor saver tomato. So flavor saver tomato is the first commercially grown genetically engineered food crop granted a license for food consumption in May 18, 1994. Even though this flavor saver tomato is not the first genetically modified crop, but it is the first genetically modified crop which is commercialized. The first genetically modified crop or plant is tobacco, but the tobacco is not commercialized. Okay. Then for the BT corn or aka transgenic corn is a corn that has been modified to produce an insecticidal protein that occurs naturally in Bacillus thuringiensis, a bacterium found in the soils throughout the world. It is naturally produced It's naturally produced crystal-like proteins that selectively kills few specific insect pests or species. Okay. And letter C, the Roundup Ready soybean is one of the series of genetically engineered variety of glyphosate resistant crop produced by Monsanto. And last but not least, golden rice is a variety of rice, specifically Oryza sativa, produced through genetic engineering to biosynthesize beta-carotene, a precursor of vitamin A, in the edible parts of rice. Okay. So BT corn, Roundup Ready, soybean, and golden rice are also genetically modified crop, but they are later commercialized. The first being commercialized is the flavor saver tomato. Question number 20. Which of the following is not considered as breeder's trial? A. Preliminary yield trial. B. Advanced yield trial. C. National yield trial. And D. None of the above. Timer starts now. Time is up. Before we proceed to the correct answer, let's define first the breeder trials. So breeder trials are conducted for primary purposes of evaluating the performance of the final set of genotypes. So that is the meaning of the breeder trials. Now let's reveal the correct answer. And the correct answer is letter C, National Yield Trial or the official trial. Both preliminary and advanced yield trial are considered as breeder's trial. So PYT or preliminary yield trial starts at early generation with a large number of entries. AYT or advanced yield trial, it is planted in fewer rows and replication and smaller number of entries than PYT. Whereas the national yield trial, which is not considered as a breeder trial, the genotype has been identified as potential cultivar and more detailed and more multi-locational trials are conducted. Okay. Question number 21. Which of the following is the last step of seed industry operation? A. Seed development. B. Seed production. C. Seed conditioning. And D. Seed marketing and distribution. Your timer starts now. Timer's up. And the correct answer is letter D. Seed marketing and distribution. That is the last step of seed industry operation. The first step of seed industry operation is the seed development. That is in the breeder's plant breeding stage or the development of the line. Next, the seed production, that is the multiplication of the pedigree seed. And seed conditioning involves seed drying, cleaning, sorting, treated, and packaging for sale. Okay. So again, the last step for seed industry operation is the seed marketing and distribution. Question number 22. Which of the following is known as the nucleus seed? A. Breeder seed. B. Foundation seed. C. Registered seed. And D. Certified seed. Your timer starts now. Time is up. And the correct answer is letter A, the breeder seed. So breeder seed has the highest genetic purity and it has white tag in the seed tagging. Foundation seed is the basic seed of the progeny of the breeder seed. The genetic priority is close to the breeder seed and it has a red tag. The registered seed is the progeny of either breeder or foundation seed and grown in seed contract growers and subjected to inspection for both field and harvest seed, and it bears the green tag, whereas the certified seed is the progeny of either foundation, registered, and certified seed. This is grown in isolation or as prescribed condition of the crop and subjected to inspection in both field and as harvested seed, and it has the blue tag. Okay. Again, the answer is breeder seed. It is also known as the nucleus seed. Question number 23. Blank is defined as the legal mechanism established to ensure the pedigree seed produced by the breeder reaches the public in its high quality, genetic purity, and original genetic identity. A. Seed testing. B. Seed classification. C. Seed certification. And D. Seed tagging. Your timer starts now. Timer is up. And the correct answer is letter C, seed certification. On the other hand, seed testing is the collective laboratory evaluation of certified seeds whose result will be printed on the seed tag. That's include the seed vigor test, seed viability test, seed purity test, and seed health test. Seed classification, as I have mentioned earlier, that includes the classification of seed based on its purity and its genetic content, and the seed is classified based as a breeder seed, a certified seed, a foundation seed, and a registered seed. Okay. On the other hand, seed tagging includes uh, the putting of tags which indicated by different colors. So in the case of breeder seed, you have a white tag, and for foundation seed, you have a red tag, and for the registered seed, you have green tag, and certified seed, you have the blue tag. That is for the seed tagging. Question number 24. Which of the following is not given consideration in designing field trials? A. Data to be collected. B. Number of genotypes to be evaluated. C. Statistical design. D. Number of season or years to be conducted or to conduct the trial. And E. Location of field trials. F. Prediction of results. Timer starts now. Time is up. And the correct answer is letter F, prediction of results. So in designing field trials, you don't have to predict the results. You have to analyze and interpret the result based on your data gathered. Okay, so letter F, prediction of result is not considered in designing your field trials, but data collection, number of genotypes to be evaluated, the statistical design, number of season or years to conduct the trial, and the location of the field trials. These are the key important things to be considered in field trials. Last question, number 25. Which seed analysis involves physical and genetic assessment of seed integrity? A. Seed vigor test. B. Seed viability test. C. Seed purity test. And D. Seed health test. Your timer starts now. Time's up, and the correct answer is letter C, seed purity test. So seed purity test includes examination of the physical feature of the cultivar seed which distinguish them from other cultivar or weed seed, and the genetic testing through isozyme analysis or DNA profiling or fingerprinting. By the way, seed purity test has two levels, the physical and the genetic assessment. Whereas the seed vigor determines the capability of the seed to emerge rapidly and uniformly into a normal seed. And the seed germination or variability test includes the rolled towel test, petri dish test, and tetrazoolium test. And for the seed health test, is used to examine the seed for the presence of pathogens by visual inspection, seed incubation, and biochemical testing. Again, that's all for this colloquium series for the crop improvement and seed selection topic. Thank you and good day, everyone.